We have a great session today. I think we're going to have a great opportunity to speak to a couple of different ideas. And I think we've heard a lot about exosomes, and we look at the molecular biology. And what we're going to try to do today is to bridge that gap maybe. What we know today, how can we use, and how can we get some confidence that we can use it then clinically? So we have an exceptional panel here, Dr. Chen, Dr. Camillo Ricordi, and Dr. Tom Temple, who will speak to us about their specific fields. And we'll go through liver, and we'll go through pancreas for diabetes applications. And then Dr. Temple, as an oncologist, will give us some idea on how we might be able to address some of the musculoskeletal tumors. So we'll have the first. Okay. So my name is Tim Ganey, and I am the Chief Scientific Officer with Vivex Biomedical. And so we have done a lot of work in the field of musculoskeletal work currently. And as I was preparing for this, I was coming down and I was trying to kind of look for one of those kind of lame conjunctions, you know, and I was saying, well, maybe it's like a stem shell conference here. And I found this piece, and when I saw that Bernie Siegel had adopted a new logo for the meeting, I said, what the heck, I'll just kind of throw it out there. But the pieces I really want to show you are going to be these pieces right here. These are these fragments, as opposed to the well-organized, the DNA. We're going to talk about the exosomes today, and these are small fragments of pieces that have amazing biologic potential. So when you look at these materials, take a look at the size. We've got the cells, and we all know about the process where you can invigorate these membrane vesicles, and they're released into the extracellular matrix. But what's really important, and I think it's a key, is to look what's inside of those. So we have DNA, we have proteins, we have messenger RNA. These are really important pieces. They do a lot of good in our body. They direct the synthesis of proteins. They affect our phenotypes. So I heard something, Alan Giacomo spoke this morning, and he mentioned David Kessler talking about these new molecular entities, and that the case and the form and the direction of a lot of the research then begins to follow these new ideas. And so I send you up a kind of a seasonal specific picture here of the constellation Aquarius. And I think this is important. These vesicles have been here a long time. When we added the ability to look through the Hubble telescope, soon we could see this helical galaxy far away. So it's always been there. We just didn't know it. And I think one of the key things that we're finding in the exosomes, how do we take advantage of the new knowledge? How do we take effect to this new ideas? How do we move forward with these ideas and basically invigorate them not only with knowledge but applications? So that's what we're going to try to go through today. So like the night sky, through Hubble giving us new ideas, so it goes, we have organs and tissue, cells, vesicles, and today our conference is going to be about exosomes. So I hope to bring you a lot of information about that. What are these biopotentials? I'm going to give a brief overview of exosomes, just kind of as give us a lexicon so we're all on the same page. We have expertise in various fields that are going to follow me, and they'll give it to their specific area. But I want to be sure to give to you just an overview here. So what are these exosomes? They're nanometer size. Think of the size of this, 30 to 100 nanometers. And when I was doing my studies, we talked about paracrine factors, and I had the good fortune to look at early cartilage factors for maybe keeping vascular support out. And they were called anti-invasion factors at the time, and then they were TIMP. And now we know that these paracrine factors are diverse and that they balance each other to actually create the biology that we know in health and in disease. But it's a cell-free regenerative potential. And I know that we have heard a lot about that at this meeting, and I think the exosomes are pretty good. They're naturally occurring. You cannot get rid of them. I've got a small cartoon here that demonstrates. So based on stress or pH or pressure or shock, you see the release of these exosomes. When these exosomes either go into the matrix or they touch additional cells, they transform these other cells. And these cells take on the characteristics of the shedded cell. They make proteins. And what kind of proteins are they making? What are these exosomes responsible for? You think of the structure of cells, and you think of the ability for these cells to be mobile, to unite, to integrate. And you find that there's signal proteins that give it form. They have the cytoskeleton. They direct the focal adhesion so that you get matrix-directed apposition. You see reduction in proliferation. You see sensations of that stimulus. And you begin to make the matrix in the tissues that you want. motion, cell cycles, anti-apoptosis. These are the things that the exosomes do. They've been well characterized. You see them throughout the literature, and I think each of these applications offers us specifics for sure, but in conjunction with the total, it's the whole that really makes the difference. So we give them fancy names, but we know what their thing is. It's about cell activity. Is there interest in the concept? Absolutely, yes. When you begin to look at the number of publications, and you look that there's more than 20,000 publications, and in any great graph, of course, it's going to be asymptotic, because as you continue to extend this timeline, you never really reach the top. We continue to add to this. And I think at some point, you reach this shift, and you change axes, and all of a sudden, what has become a rise in publication changes, and you begin to mark these things. What are they? What can we be using them for? When do each of these pieces then assume their own area of expertise, and where do the people find that they can use them? So in terms of stem cells and mesenchymal stem cells, this is an interest here. So there's awareness, and there's evidence, and the key thing is, what do they do? So we found that they protect, we know that they stimulate, we know that they give gain of function. So within this stem cell summit, I think the individuals who are attending here are looking for that regenerative potential. And so the mesenchymal stem cell has been a great base. We've heard about that for many years, but I think the applications are really the key. And where are we in those applications? What have we done so far? So the evolution of interest, there's 20,000 publications on exosomes. But here's what really kind of strikes me as so unusual. It's all academic. Where are the funding dollars? Where is the evolution of this idea? Where do we get it to the clinic? And how do we avoid what we've heard all through the day is not only being speed bumps, but walls that we have to conquer? And I think that's some of the work that we're trying to do. And I think the leaders in this field that are assembled today to speak to these will give you some ideas on what those challenges are and what they're doing to get around them. It seems mandatory in this meeting to at least go back to clinicaltrials.gov to get kind of a gut sense check. How much of this is being tested? How much is out of the academic? How much is going into the mainstream? How are we able to actually to enable ourselves to know more about how it does in people? I've heard many stories today of irradiated mice and irradiated monkeys and lethal doses and potential recoveries. But in the end, it has to go into all of us in this room, those people who have had some effect with health. And I was glad to see as I looked through these 68 trials, eventually I got to 2 of them that were actually looking at healthy people. What's normal? Let's not look what's in disease. Let's find out what's healthy. And then let's move the direction to maybe accommodate that and find some resonance, that allows us to achieve that health. And so of these 68, only 10 have been completed. 3 are terminated without finishing. And then there's 55 more that are still recruiting. So we're right in the beginning of this. This is the nascent science. We're in a really good place to not only know, but to learn. And I think it's really an important place. I'll give one more quick cartoon here. To go over this again, it's important. So when you give cells, they have these exosomes within them. And you could have a recipient cell. So let's say I'm donating some of my cells, or I'm donating some of these cells for possibly use in another application. The recipient has the ability to pick up these exosomes, and it tends to go back to the donor. So you have the ability not only to accept the exosome, but also accept the quality, the phenotype, and the functions of those cells. We've been doing this in the laboratory, and this is some great work that has been done at Vivex. We feel very strong about this. So we take a mesenchymal stem cell population just as a starter, and I'm using this as an example. So we take one, we stain one with one on the cytoskeleton. We do another, we stain the membranes with a DIL stain, a different type of membrane stain. But what's important is that when you mix these cells 3 days in culture, you find that you're getting an exchange of the intracellular material. So for many years, we've been giving allografts, and Dr. Tom Temple, who's one of our speakers, was the director of the UMTB for many years. And of all the allografts, he brought this to my attention. He says, we've been doing this for a long time. This is not new science. This is just defining the science. This is recognizing it. This is just giving it a new name. And so what we're trying to do with our group at Vivex is to develop a way to do this carefully, controlled, and be able to kind of annotate the dimension, the detection of these cells with names. And that's what we're doing. What you find when you bring these cells together is they don't lose their phenotype. So if you have something that's mesenchymal in origin with a CD90, you find that even as you exchange the exosomes within them, you're able to retain the functional characterization of those cells. We did this a little bit more. So we have a product that's based on a select group of Miami cells, and they're selected mesenchymal cells. And so we said, well, once we've separated these, as opposed to just having a passage cell from Lonza, do they still do the transfer? And so I think that's something we really wanted to take a look at. So we did the same thing. So we did a staining on our cells from Lonza, mesenchymal stem cells, fifth passage. We did a DIL stain on the membrane. And I call your attention to this because I think it's really kind of cool. So in the end here in the bright field, you can't tell the difference. The cells all look the same. But when you merge the cells and you look at the fluorescent aptitudes of these, you find that you've actually done the exchange. So we're able to take the select cells that we have with a high phenotype towards a primitive type of mesenchymal cell and attach these to the mesenchymal stem cells that are typically a fifth passage from Lonza. Another picture, same discussion, that you can't tell the cell by its color until you put some color on it. So we're targeting right now a translational project. We're looking at intervertebral disc. We're using nucleus pulposus allograft. We have a biologic component that's a preserved cell, which we keep frozen so that we can actually give its potential when we connect it to the allograft and do a delivery. It's all percutaneous. So our gain of function in this exosome transfer, the goal is to take a very primitive phenotype that has many potentials and to offer this as a way to the nucleus pulposus cells and to then allow those cells to accept that more primitive space. And if you look at the nucleus pulposus, and many of you have done, I think, a lot of the embryology, you recognize that the somite segmentation is a big part of our spinal development. You see this segmentation of somites, and you see them all clamped together. What remains is this nucleus pulposus. And what we find within ours is that the nucleus pulposus cells are very primitive. And so we have a high proportion of those somite-specific antigen in the stem cells we select. We also, and again I go back to Dr. Temple in appreciation for this, when you begin to take the nucleus pulposus material as an allograft out, or if you take a micronized bone or a micronized cartilage or micronized skin, you find that you still have cell components. If you look on the left, you have a DAPI stain, which recognizes the nucleus, the material that we're doing. There's still cell components in. There's also, if you do a PI, you recognize that all these cells are dead. Certainly in the cleaning process, you may have taken some of the vitality out, but you've certainly left some of the cell markers inside. You can also find that within these biomaterials, within these carrier molecules, you're still finding exosomes that are present. So we're finding that we have an allograft material of nucleus pulposus, certainly is capable of engendering some vitality in the cells that we're putting in, and that there's an absolute exchange of the material between the cells given, the cells carried, and the host cells that are getting the grafts. So in this regenerative spine care, not only are we giving the cells, but we're giving those ghosts. We have the ability to put those ghosts of the exosomes into the material, and we're putting this into the disc right now. So we have a clinical trial that's underway. We're looking at this as an HCTP allograft. We're supplementing tissue. We're protecting the cells, and it's a percutaneous type of infusion. So the summary of that strategy, I think, is a key, and I'll underscore it second here to close. But basically, it's a regenerative intervention. We have a percutaneous delivery. It's an allograft material. It's 361 regulated. It has a viable biologic component, and we have those non-whole cell paraffin fractions. So what this is a very, very, not a naive approach, but it's a very basic approach. What we're trying to do is capture the ability of those cells to enliven the grafting material, to give some credit to the particular parts of the grafting material that's functional, and then to demonstrate that we can do this in clinical patients. So the future for exosomes, I think, there's immense here, and I won't dwell on these. I think there were a lot of sessions, and we can have this discussion later. But I think, with liquid biopsies, being able to determine in particular, what is the prevailing issue of the disease state so we can get back to the health. You can use acoustic microfluidics to separate these, point of care for taking exosomes out. Precision medicine. I heard a great story here on the cord blood that I was impressed with yesterday. When you begin to look at Excelthera and you see abilities to actually synchronize the ability to use the donors, particularly in a cord blood, as opposed to having a one-to-one reciprocation of need, and you only get to use each one, be able to use those genomes, be able to expand them, almost like an induced pluripotential basis, but using stuff that has been given, donated, and is no longer used. So, with no further ado, we want to go into our program here. We have three great speakers. Dr. Chen is going to lead us off from the University of California in San Diego. She's going to speak on liver dimensions and exosomes. Dr. Camillo Ricordi will follow. He's going to talk about diabetes, exosomes, and being able to give gain of function towards cells that can reduce a lot of the insulin dependence. And then Dr. Tom Temple is going to close, and he's going to speak about some of the challenge of translational medicine, the ability to use the musculoskeletal oncology to maybe attack those tumors in a very specific way, knowing what the sources are. So I open this up to Dr. Chen as our next speaker. What I will do is call the speakers back to the podium at the end. It's difficult to see from here. You'll end up with a slanted opinion. And then when we've had everyone had a chance to speak, we'll bring the speakers up, and we would certainly love your questions. Thank you.
Yes. Thank you very much. And it's a great introduction for exosomes, and also it's a wonderful work. So I'm following for this topic for exosomes, and I will talk about the exosome application in liver fibrosis. So this work is lead in the liver fibrosis research field right now. So at the beginning, as we all know for liver fibrosis, so liver fibrosis is a common pathogenesis involving excessive deposition of scar tissue that is rich in collagen. And the liver fibrosis causes the loss of normal tissue structure and function, and that leads to the disability and death. Unfortunately, till now, fibrosis of internal organs is a significant contributing factors in 45% of the United States staff and affects millions of people worldwide. The main causes of liver fibrosis are alcoholic liver disease, hepatitis, non-alcoholic fatty liver disease, cholestatic liver disease, as well as other liver diseases. Despite the magnitude of this problem, there are now still lack of the methods for diagnosis of liver fibrosis, and also there are no FDA-approved antifibrotics. So next let's get closer to look at the process of liver fibrogenesis. So in response to the chronic liver injury, the main population of the liver cells, hepatocytes, get damaged and lost their function. At the same time, another important group of cells, hepatic stellate cells, as I shown here in blue, become activated and differentiate into the myofibroblast-like cells that produce wound healing and pro-fibrotic factors, which drives collagen deposition and scar formed. So this is the process of fibrogenesis, and during this fibrogenesis, the hepatocytes, stellate cells, as well as other liver cells, they communicate and they exchange the information. And it has been recently found there's a new organelle called exosomes get involved in this process and transport the important biological signatures between these cells. So let's talk about what is exosomes. Well, we have heard in the previous talk the details of that. I will introduce a little bit more about this. Exosomes are nano-sized vesicles. Their size between, let's say, 30 to 100 or 50 to 150 nanometer in size. They're really small. And they have a common set of membrane and cytosolic proteins. They contain a complex molecular cargoes consisting of cell type specific proteins, messenger RNAs, and microRNAs, which reflect the origin of their producer cells. An important function of exosome is to act as molecular shuttles. Following their release from donor cells, exosomes cross the intercellular space and deliver their contents to the recipient cells, which are then functionally reprogrammed. So these exosomes not only transferred between the cells, they are also present in the body fluids, including blood, urine, and saliva. So because of this, these exosomes in circulation could be a readily accessible and rich source of biomarkers that have high potential for assessing the organ diseases, including liver fibrosis. So that's what we are doing now, to test this possibility and to identify the signatures of fibrosis in circulating exosomes. We first used a 5-week carbon-TAT fibrosis mice model, and we screened 940 microRNAs in these circulating exosomes from carbon-TAT fibrosis mice compared to the normal mice, and we confirmed this change by real-time PCR. As you can see here, the series red staining showed the collagen accumulated in the liver in the carbon-TAT injected mice, which proved the fibrosis model. And this heating map result showed the change of microRNAs in the circulating exosomes between the carbon-TAT mice and the normal mice. And you can see the red color represent the upregulated of microRNAs, and the green color represent the downregulated microRNAs in the circulating exosomes. And on the right side, I listed the most upregulated and downregulated microRNAs. They are fibrosis-related. So based on this, we continued to screen the samples from human beings. So we collaborated with Chinese hospitals. We get about 6,000 patients, and we selected, based on the criteria, pull some of the samples. They are from the fibrosis progression, or they are from regression groups in HBV patients. We compare them with the healthy people. So this is a heating map. You can see. In this heating map, I want to emphasize 2 things. The first thing is the first line you can see, that is the stage 3 fibrosis patient samples. We definitely can see the change of the microRNA levels in the circulating exosomes. And very interestingly, some of the microRNAs, which maybe you cannot see here, they are very conservative, and they show the similar result as we compare to the mouse screening results. So, however, in the second line, in the fibrosis regression group, and this group of patients, they already reversed, getting close to the healthy level. And in this group, the microRNA levels almost showed no change. So this result tells us these microRNAs, the change of these microRNAs in circulating exosomes, they are tightly associated with the stage of liver fibrosis. And from this, these results suggest the profiling of circulating exosomal microRNAs have highly potential for diagnosis of liver fibrosis. So I only talk about the exosome microRNAs. We definitely can do the proteomics and to provide the proteins and to get more signatures for fibrosis as well. So after this, I want to catch your attention on the downregulated microRNAs I listed on the right side in green. And another way of viewing this data is the reduction of these microRNAs from the baseline levels in fibrotic mice represent the loss of microRNAs that serve a protective function in healthy mice. This is hard to understand. Let's say a simple way, use a simple way to think of that. It's the exosome produced by the healthy cells. They carry the therapeutic signatures. So the question is that, can they reverse the fibrotic liver injury or not? So to do that, we come back to the animal model we used before, and first, we stained the circulating exosomes from the normal mice with a fluorescent PKH26 dye, and we inject them back to the fibrotic mice 4 hours later. We take out all the organs from these animals and image them. This is the result. So the fluorescent signals exclusively localize in liver, but not with other organs. But this is only 4 hours later, right? And then with the same injection and the isolation And the imaging result shows that these fluorescent signals mainly localized to the hepatocytes and the stellate cells, especially they dramatically increased when these cells got injured. So this result shows that the circulating exosomes from the normal mice, they can homing back to the liver cells. And next, we want to ask if these circulating exosomes are in fact antifibrotic. So we go back to the fibrotic animal models we used, the steel carbon tat injected for 5 weeks, and we injected back with circulating exosomes every other day during the last 2 weeks of these animal models. And we can see, as expected, the carbon tat increased the accumulation of collagens in the carbon tat mice. However, with circulating exosome treatment, we saw this increased levels of collagen almost disappeared, and the liver recovered. So these results show that the circulating exosome are indeed antifibrotic. So this is just what we have done in the experimental liver research. So summarize what we have seen is that the profiling of circulating exosomal microRNAs, messenger RNAs, or proteins have a huge potential for diagnosis or prognosis of the liver fibrosis. And also the circulating exosome from healthy mice are therapeutic for experimental liver fibrosis. So I would like to say the circulating exosomes from healthy individuals may be a platform technology for treating like liver fibrosis and also other fibrosis in multi-organs, as well as other diseases. So thank you very much. And I will say we will never lose the hope to fight for the liver fibrosis. Thank you.
Thank you very much. That was exceptional, and I like your never give up, keep that hope alive. Our next speaker is Dr. Camillo Ricordi, and he was recognized earlier this morning in a session. We're looking at the real leaders in this field, one of the stem cell giants, and we're anxious for him to step up and give us some insights into exosomes and maybe future of diabetes research. Thank you.
Thank you. Thank you for providing the pointer. It's actually it's good that we have already some introduction on exosome biology and function, so I will skip these introductory slides that are repetitive, but just to mention how it is a new mechanism of stem cell to host signaling through exosome that adds to the well-known other mechanism, paracrine, content-dependent, or endocrine-mediated signaling. And our interest in exosomes started when we observed some of the emerging literature on effect at a distance from cell-cell contact when you inject MSC-containing cell product. And we started looking specifically when you use enzymatic digestion versus non-enzymatic digestion, whether there are differences in the exosome content or delivery to tissue, so that our fellow Marta Garcia Contreras, who did most of this work at the Diabetes Research Institute, look at this difference between like lipogen-derived or enzymatically processed fat-derived MSCs. And it was interesting, as I mentioned before, to see that there is a huge difference in exosome content, but the interesting thing to us was also to start characterizing this lipogen-derived MSC exosome for growth factor-related components. And this is too detailed to go through also because I cannot read anything from here. But as you can see, there are big differences if you go from lipoaspirate to lipogen-derived cell product. And this is for angiogenesis-related growth factor and product. This is for growth factor-related messaging. So in summary, you could say that basic fibrovascular factor FGF and FGF2 were both significantly differentially expressed. And to use MSC exosome therapeutics in type 1 diabetes, we want to see if we can improve the function of transplanted islets, avoiding primary non-function, but also support the early loss of islets that can follow transplantation, since you put microorgans in a site that is not immediately vascularized, and you have to wait for new blood vessels to grow to provide the nutrients and the oxygen necessary. So we started characterizing exosome from adipose-derived MSCs, and it was interesting to see that if you use like adipose-derived MSCs from lipogem or the condition medium from this MSC that doesn't contain exosome or MSCs alone, you can see that you have migration of exosome. You don't have in the condition medium that doesn't contain exosomes, but you have migration both if you have MSCs or purified exosome from that same condition media. So that proves the migration inside the islet cells, as it has been shown before for the liver. We also were able to see that you could improve insulin secretion in response to glucose, glucose-stimulated insulin secretion, if you both mix islets with MSCs, but even more if you mix islets with purified exosome from those MSCs. And these are early findings. The other thing that we were very interested is to show if you can prevent the hypoxic damage and loss of islets. That is something well known when you do kidney capsule, renal subcapsular transplant in rodents. You can see if you do a trichrome stain, you can have a history of what happened early. So if you look at the transplant 200 days after, you can stain for insulin and say, great, we have engraftment everywhere, full thickness. But if you look at the collagen and fibrosis deposition with the trichrome stain, you see that when you're close to the renal cortex that has the same kind of oxygen tension of native islets in the pancreas, you have full survival and no loss. As you go towards the peritoneal cavity and the islets have to get revascularized from these blood vessels, then you lose maybe 60% of the tissue. When you do tissue engineering graft now, engineering a mini endocrine pancreas in the omentum, in the abdominal cavity, you see that this loss is full thickness. So we try to see, can you now improve survival of this tissue while you provide angiogenesis? And that's why we're so interested in using this microcluster of adipose tissue or stromal vascular fraction or MSC and endothelial products that could be tested in this direction. So the reason we like particularly the microcluster of adipose tissue is that, to me, this could provide a factory of exosome or MSC that is sustainable. It's not just that you have to continue to inject exosome every day, but possibly you inject a system, a microsystem, a niche, maintaining the stem cell niche and the factors that could secrete them. So can this work? Like, this is a crazy experiment that Carlo Ventura did in Italy, because when he told me that he was doing these experiments, I said, forget, you're wasting your time. First, you're putting human cells into a rat, and in our experience, within 2 days, everything disappeared because it's xenorejection. And second, how can you prevent ischemic injury if you ligate the femoral artery? You don't have time to rescue the limb because it will take days, weeks to have full revascularization. So there is a very important message in this slide, because not only you maintain human fat microclusters in a rat for weeks, but you also have survival. You reverse ischemic injury before the blood vessels grow back, and you have the new blood vessels that rescue the limb. So it's probably a dual effect that may be exosome mediated. In this case, there are no specific staining for exosome. But the fact is that you may protect the tissue while you promote angiogenesis, and that could explain. The other thing that triggered my curiosity is, say, wait a minute, if we put islets from human into a rat and they're destroyed into 2-3 days, you find 0 cells. And now you put this adipose-derived cluster with MSC and exosome, and they're still there weeks after. Maybe there is also an immunomodulatory component of this exosome or the MSC or the stem cell niche or whatever that could be useful also to maintain islet transplants in an ectopic site. So that's one of the reasons that we started looking more into this. These are part of the characterization of the angiogenic paracrine factor secreted in 48 hours in co-culture of islets with MSC-derived exosomes, and basically it's just the beginning of something that I think will increase as interest both in our field and many other fields, as you saw brilliantly in the introduction, just looking at the number of papers and the number of clinical trials that are now beginning. So thank you very much. Thanks.
It's a wonderful application. It's always good to see some kind of novel finding, you know, and when you are concerned about that viability in that rhythmic rat model and you see that support, it drives your thinking down a different chute. And I really kind of impressed you know, you're being so candid with us and your expectations. Thank you. Our next speaker is Dr. Tom Temple, and Dr. Temple is he comes from a lot of different areas. Currently, he's Senior Vice President of Translational Medicine at Nova Southeastern. He comes from a host of experiences and skills. He's going to speak about the clinical utility of exosomes for us. Thank you.
Thank you, Tim. After seeing such extraordinary science, it's a cruel irony to have an orthopedic surgeon opine on the value of exosomes and therapeutics. So we've all heard about what exosomes are, and I won't get into this. What's in the exosomes, proteins, lipids, and nucleic acids. How do things get taken up by other cells, specifically exosomal materials, either intracellular signaling, the paracrine effect has been discussed. RNA proteins are transferred selectively, and finally the entire exosome may be phagocytized and the contents therein taken up by the cell. What do exosomes really do? So they present antigens, for example, dendritic cells of the host and recipient. They regulate the immune response in both homeostasis and as well as dysregulation in patients with cancers, for example. As Tim Ganey showed, that they participate extensively in tissue repair and regeneration. They facilitate the spread of infectious disease, and of course they have epigenetic factors, and they act as promoters. In my field, we haven't done all that well surgically in controlling many of these sarcomas. This is an example of an individual who had a myxofibrosarcoma that's recurred multiple times, and you could see that the intervention for this is sometimes quite extreme, and the results are not as satisfactory as we might like. We have many cases in which we have absolutely no solution to bad problems. In this particular individual with a differentiated chondrosarcoma of the femur bone, and you can see that the tumor is extending up and down the bone and within the bone in ways that are simply not amenable to a therapeutic intervention, requiring in many cases major ablative therapy. So it brings us to the biggest problem that we have, and that's controlling metastatic disease in many of these patients with soft tissue sarcomas and sarcomas of bone. Metastatic disease to the lungs and to the bones, and principally this is the cause of death in many of the patients with this particular disease. So how can we exploit tumor-derived exosomes to diagnose and mitigate some of the effects of cancer? And certainly what we try to do is we affect the cancer growth along many different stages, whether it's the tumor-stromal interaction, the migration of these cells through the matrix, invasion in distant sites, eventual engraftment, and then the immune modulation that occurs. and somehow being able to retool these exosomes to determine, number one, the response to therapy and actually affecting clinical response to therapy. So the first way we could exploit exosomes is using them as biomarkers. And what we do know is we can extract exosomes from tumor. We can actually measure them. We could measure them in normal tissue and in normal blood, and we could look at the amount of exosomes in patients with cancer. And you could see that there's a multifold increase in exosomal RNA in patients who have cancers. So we could actually look at these exosomes in healthy individuals, and we could develop a exosomal profile for patients who have tumors as well. And then we can think about how these signatures change in patients with both malignant and benign disease. And in patients who have malignant tumors, we could look at those that are at risk for metastases and those that remain localized or in situ. And finally, when we think about metastatic disease, we could look at, number one, tumor aggressivity, the effect of drugs on the tumor and resistance, and finally, treatment responses. And there are some examples where we have used exosomal material to basically think about patients with pancreatic and breast cancers as well as prostate disease. So exosomes really do stimulate tumor growth. They're involved in tumor invasion as well as metastases. And by inhibiting tumor exosome release, we may be able to exert a therapeutic effect in overall cancer therapy. And this has been shown by Kasaka and his group, where you can chemically block ceramide synthesis, and this seemed to have an effect on tumor exosome release. The concerns, obviously, are if you have systemic off-target effects by inhibiting exosomal release, and these may have undesired effects on the inhibitor itself. And finally, is the inhibitor tumor-specific, or does it have local and systemic effects? One of the ways in which we can exploit exosomes is through vaccines. And although some have shown some benefit with vaccine therapy, we haven't seen substantial improvement in local disease recurrence or survival with sarcomas. But the idea is to culture these antigen-presenting cells along with the tumor cells, extract the exosomes, and then inject these into the patient with specific malignancy. The dendritic cells are presented with the antigen, and they incite a T-cell activation, and this T-cell then is used to incite an immune response to the tumor cell. The APCs, number one, are very difficult to culture, and very specific tumor antigens have to be transferred to these APCs. These also require an MHC1 haplotype matching, so that each preparation has to be prepared individually. Another way to use tumor-derived exosomes is to basically culture the tumor cells, extract the exosomes, and inject them. Again, these go to a presenting APC, in this case a dendritic cell. Again, a T-cell response that incites activation, an immune activation in the tumor. And this has been done with melanoma, as many of these trials have been shown to be effective in melanoma, and to a lesser extent in non-small cell lung carcinomas, not so much in sarcomas that talk about a little bit. Another way to exploit exosomes in the treatment of neoplasia is to heat these tumor cells and then to express these exosomes that are associated with heat shock proteins. This is an old iteration of a concept of Cooley's toxins that have been shown to be effective around the turn of last century. These exosomes are collected and transferred to the recipient, and again, they stimulate monocytes and dendritic cells, which elicit a T response, and that could be amplified to attack a specific tumor antigen. They can also directly activate NK cells, which we have a specific interest in our laboratory at NSU. And these NK cells then could be initiate apoptosis in these tumor cells. So this is an area that is quite interesting to us, and is actually a scalable process. Finally, we can create a competitive implant. So the exosomes are embedded in a 3D scaffold that can be implanted in the human body, and these will compete for tumor explants. And this material can then be excised and decrease tumor burden, and this has been done, for example, with certain ovarian cancers. Exosomes can also be designed to carry biologics or natural and synthetic liposomes, perhaps in mimetics. Here we could take a chemotherapeutic agent. We can directly package these exosomes by electroporation and inject these and induce toxicity within the tumor cells. And here we can actually amplify the effects of Paxil in lung cancer. Not so much with doxorubicin and ifosfamide in patients with mesenchymal tumors. These can also be collected from transfected cells and can cause direct macrophage activation toward an anti-tumor phenotype. And this has also been used with some efficacy. So finally, when we look at the challenges of using exosomal strategies in treating malignancies, the isolation is very expensive and it's time-consuming. It requires an enormous amount of technical expertise and variability between technicians, as well as variability in our case in sarcomas between tumors and the host. In fact, many of these sarcomas within the tumor itself have extraordinary genetic variability, and we've been actually able to identify up to 5 different phenotypes from a single tumor in a malignant peripheral nerve sheath tumor, for example. So how do you take that heterogeneity and consolidate it into a cogent strategy to treat a tumor? We need to minimize co-isolating protein aggregates, for example, albumin, and all steps of a single protocol have to be optimized for greater efficiency. The ability of healthy and diseased tissue and serum from patients over time is sometimes a logistical nightmare and may not be possible in some cases. In our particular instance, we recover the tissues in the operating room. We begin the process of cell preparation before we even take it to the lab, and then the lab, of course, is extremely responsive and begins the preparation seeding for future culture purposes. So the solutions are that we need to develop a pipeline that minimizes effort, maximizes adherence to protocol, and ultimately is very efficient in the way its tissue is collected and finally processed. We need a system that's optimized at every step, and that involves a very tight protocol and SOPs that are not only specific to the patient, but the particular tumor type. These need to be patient-centric applications that could be applied to larger groups of individuals with similar disease profiles to produce the scale, optimal range, and acceptable cost, and this is all going to be quite complicated by what seems to be a certain regulatory morass in developing these kinds of strategies. So really, fundamentally, bench-to-bedside approach requires a multidisciplinary team of individuals on the clinical and research side, and what appears to be the need for an extraordinary computational biologic strategy where we can manage these exceptionally large databases and informatic platforms to basically develop cogent signatures for specific tumors, tumor types, and then somehow individualize these to patient care to improve outcomes and extend life. So thank you very much.
That was exceptional. We have some time for questions. I'd ask the other presenters to come to the stage if they would, and we'll sit here as a panel. And I'm just interested in several questions that I have personally, but I open this to the audience. As you have questions, just raise your hand, and we'll certainly acknowledge that and ask you to speak up and offer this to the whole group of participants. As people are kind of formulating their questions, let me start with one. I'll start with Dr. Chen. And I was amazed to see your fluorescent labels, being able to demonstrate that the exosomes in which you had implanted had not only nourished the cells, but actually gave some recovery of function. Mm-hmm. When you look at those cells that have recovered, do they, and again, then almost like a domino, they begin to create exosomes that further the success within the repair? Do you see them making exosomes that are actually for healthy cells, or do they continue to show degrading type?
Yeah, so to simplify your question is about all this labeling of the
yes,
exosomes.
so you did a good job labeling, and you showed where the exosomes were in the recovering cells.
Yeah.
Do those cells, once they've received those exosomes, and they show the fluorescent markers, do they then begin to show healthy tissue type secretions themselves?
Yes. So when we have this labeled exosomes back after injection, we also isolate these cells, and then these cells, all the phenotypes, for example, like hepatocytes, they got more proliferation rate, and for stellate cells, they definitely inactive, and they have less collagen and alpha-summate like activation markers, decrease all this expression. So all these cells labeled, we compare to before and after, and all these liver cell types, so they reverse back to normal.
Very good. Yes. Very interesting.
I was, if I may ask a question, I was fascinated by your data that you see more penetration of exosome when you have injury. Okay. And this could explain also, like in early work, like when Claudio Bordignon decades ago was doing testing of bone marrow transplants in muscular dystrophy or in muscle regeneration, you see that if you didn't crush or damage the muscle, you didn't have an effect of the bone marrow. And in those days, we didn't look at exosome, but I'm wondering if it is a common pattern that if you have injury in any tissue, you may have more mediated effects through exosome and microvesicles because they can penetrate more easily, probably.
Yeah, definitely. This is a great question. And we also see, well, it's not our work, from the literature, so definitely the exosomes can look like, or they can target for the macrophages, and also Kupffer cells like in the liver, and they also function to for the inflammation process. And for us, the point I want to say is that our circulating exosomes, we tested all the inflammation cytokines, and they definitely reduced all the inflammation process. And another point is that all these circulating exosomes, they won't have any side effect for the normal tissue, for the normal animal model. Yes. So for stellate cells, when they get activated, they definitely differentiate into the myofibroblast-like cells. And they are different from the fibroblast cells, definitely. But so far in the fibrogenesis process, they both contribute to this fibrogenesis. And well, stellate cells are still the main population. So when we try to use exosome to cure for this disease, and then we definitely see the significant reverse of the liver fibrosis. Yeah.
One for Dr. Temple. So, thank you. Thank you. So Dr. Chen, I was intrigued by your concept you mentioned at the end of your talk about using blood product from normal donors and deriving exosomes to treat cirrhosis patients. So practically, do you know whether or not storage of the blood will change the exosome pool and therefore change the therapeutic potency? Would you have to isolate the exosomes fresh, remove the white cell component? How would this work?
Yeah, thank you for your question. So far, what we have done is we definitely use fresh isolated serum exosomes to treat the animals. And for the storage, right now we still need to figure out. So far, what I know is that for 6 months, storage back to minus 80 shouldn't affect for the quality of the circulating exosomes. But for longer time, we'll still need to work on that. But so far, I think freshly isolated circulating exosome, the best way to do this in the experimental liver model. So.
Thanks. And one question for Dr. Temple. So David Leiden, I believe, published 12, 13 years ago a very provocative paper where one could take conditioned media from, say, I can't remember if it was a breast cancer line or a a prostate line that would metastasize to bone, and prime the animals with this, and this would then direct metastasis of melanoma, not to the lung, but to the bone. So thinking about that strategy and exosome-directed metastasis, have you given any thought to trying to exploit this to maybe minimize metastatic flood after surgery, or minimize metastatic spread by directing metastases to a sink or some type of a filter type concept?
I think really two very interesting strategies. One, identifying exosomal profiles, first of all, and trying to understand what the individual exosomal fractions really do. Do they increase tumor growth or tumor movement through the matrix? Do they prevent invasiveness at the target organ, and finally tumor growth at that site? What are the factors involved? So we have an idea of what types of exosomes are being expressed, but really not a very good understanding of, one, which fractions are important, and two, how to deliver them in an optimal way. The second, using a sink, is compelling, but maybe just a way of reducing tumor burden, but not really eliminating metastatic potential at other sites. So it would be a competitive sink in a way, but really not one that, in my view, would be a good long-term strategy. However, if you did have a competitive sink and you were able to cytoreduce metastatic deposits, that may provide opportunities for other conventional strategies and/or checkpoint inhibitors. So in a way, we think of not cancer cure, but cancer maintenance, and strategies to sort of create a chronic disease out of one that can be acutely disabling and morbid.
A question for Dr. Ricordi and the panel in general. In the situation of acute ischemia, we know that muscle is sensitive to damage within 24 hours. We also have seen as early as 72 hours that SVF in diabetic ulcers can induce vascular changes, but you've got a 3 to 5 day window for vessel proliferation. Any ideas as to what mechanism muscle can be preserved under acute ischemic conditions with the transplantation of MSCs? Does it have anything to do with mitochondrial stabilization? How in the world does muscle survive?
Yeah, it's actually is a very interesting question. You may respond in the first row, but so far we just observed the phenomenon that to me is fascinating. Both is raising more questions than answers that we have so far, because we are maintaining the tissue before new blood vessels go. These were done at 2 weeks after the control animals lose the limb, and the treated one are now beginning the angiogenesis revascularization, but in the meantime, you maintain the tissue. That to me is spectacular. And also, the other surprising thing is that you find human tissue in a rat, so that implies some form of immunomodulation that I would not have predicted to begin with. But it would be very, very interesting to study more into the mechanism and what are the exact components that are responsible for this double effect, if you wish. And you may have the answer already. No?
No, I don't think so, but Keith March, Florida. My question to anybody, but probably particularly with regard to the liver finding, is that there have been a number of reports relating to anti-fibrotic activity. And in fact, I think my understanding of what you showed is not only anti or prevention of fibrosis, but actually reversal. Quite remarkable. We have some data that was parallel, we have not yet published, but in a kidney model of altered renal perfusion, which we've seen happening in the context of smoking, that if we provide secretome after establishment of renal fibrosis, in which you see both reduction of absolute flow or perfusion and clear histologic changes, that within several weeks after secretome provision, this is reversed. I mean, to me, it's unprecedented. Your data, I may have missed something for sure, but your data is the first I've seen recently, or maybe at all, that looks like that. And so we have no idea yet, well, I guess I would say, what the mechanism is, and I wondered if you have any idea, how can this be? What is the mechanism? Because fibrosis, instead of normal healing, is obviously such a huge problem in so many tissues.
Thank you for your question. Then this is a very challenging question. So for the mechanisms of those exosomes to reverse the liver fibrosis, it's a really, I should say, it's a really multi-signal pathways get involved. And I want to just mention one thing we have done for the receptor. So we have figured out some receptors can help to uptake this either circulating exosomes or the cell-derived exosomes, and especially when the cells get injured, for example, like stellate cells, they express more integrins, and then they will help uptake of these exosomes and get the information from the exosomes. This could help the exosome uptake and to function into the cells. This is only one part of our research, and for the other mechanisms for the function, there definitely need a lot of work on it. So this is all our studies for right now.
So you don't know yet what the
Not
mechanism
yet, not yet.
We also.
Yeah, we just see the readouts, but it's still very hard to link. We have all the pearls, but we cannot get the necklace yet.
Thank you. Anyone else have insight into that?
No, but I know that probably we are all hyper-secreting exosome right now because of freezing stress of near cryopreservation temperature in this room.
I would send it back to Dr. March as well. What is your kind of guiding philosophy or your mechanistic approach to that? How do you feel that you overcome a fibrosis or a laying out of collagen or an insulation of membranes? You know, you see a very neutral membrane charge, whereas in a normal type of a liver, you're going to see a standing voltage across your membrane. What's guiding fibrosis from, are the exosomes changing? Are they inserting blocker molecules into the membrane? Are they changing the polarity?
That's a really great question. I don't know the answer, and I've actually not been thinking in that way, because honestly, until we saw this data a few months ago, and then actually I was in the middle of a move, so we haven't had the opportunity to think deeply about it.
Yeah.
I just found it to be very remarkable. Generally, what we've thought mechanistically is that we are introducing a host of paracrine factors, perhaps facilitating their entry by virtue of presentation with an exosome. I think that's likely to be the case, but really what I don't really have an insight into it, but I have not looked at membrane potential in any regard.
Okay.
We do a fair bit of cardiomyocyte work, however, we haven't done entailing of the membranes. Did you ask specifically?
Well, that's why I know some of your history, and we've done some work in the past looking at pulse electric fields and looking at epithelial migration and looking at the ability to kind of override prostaglandin and nonspecific COX inhibitors using electric field dynamics. And it just struck me as one of the very interesting areas here is membrane insertion, kind of like, you stick your finger in a wave and you get a different pattern. And so almost like a peg in the edge of the membrane that really just gives you a different transmutation of that membrane potential, and so you don't make the same type of proteins you would. It's a fairly simple idea, but I think it's a challenging test, basically.
It's very interesting. Thank you.
Yes. Okay.
Wait a second, hold on.
Yes, I have a question for the panel. Thank you for the great presentation today. And also, my question is cancer therapy with exosomes are being implemented today in hospitals?
I couldn't hear the question. Cancer?
Yes, thank you for the great presentation.
Yeah, that I understood, but was the second part. Thank
yes,
you.
Is cancer therapy with exosomes now being implemented in hospitals in the United States today? Or?
So only on IRB and investigational protocols, to my knowledge, they're certainly not accepted as standard care, and they're certainly not reimbursed under any third-party insurer.
Yeah, one area we didn't discuss is also in wound healing and this kind of application, because I didn't talk about this because we focus on diabetes in this talk, but we also have data on if you do the in vitro scratch wound test on epithelial confluent cells, that the actual effect on promoting wound healing that you see when you put MSC or MSC conditioned media is mediated largely by exosomes. I know that there are clinical trials that are going to start in this direction, not from our groups, from dermatology groups, but is another area of interest, like wound healing, burns, diabetic ulcers.
I was just going to mention in discussion of the mechanisms for maybe specifically hepatic cellular uptake and things of that sort. There's so many facets that are involved in the creation of the exosome and in the transport and in the uptake, as we were talking about earlier, that once those things are sorted out, then we can look at the creation mechanism as it directly relates to the uptake mechanism and like ubiquitin and lipid mediated and things of that sort.
Another question.
I have a question about the fibrosis project. Did you have the opportunity to subcategorize the serum-derived exosomes depending on the cell type or lineage that they are secreted from, and then see their specific impact on the regeneration?
Okay, thank you. So your question is about the cell types in the circulating exosomes that contribute to the fibrosis therapeutic, right? Yeah, actually, the answer is that we definitely can sort out the hepatocyte exosomes from the circulating exosomes. And also, parallelly, when we do the other exosome therapeutic from the hepatocyte exosomes, we do exactly the same in vivo study, and they show the similar results. And there's a hard part is about to sort out all the liver cell types exosomes, because it's very hard to figure out but the markers for all the cell types, especially for stellate cells and some other small cell types. Yeah.
And maybe one interesting question on the fibrosis is like, is the anti-fibrosis effect due because you better preserve tissue? Like, is the fibrosis subsequent to cell death or tissue injury, and then fibrosis is a reaction? or is a pure antifibrotic effect? Because if it is a pure antifibrotic effect, maybe you would have application also in scleroderma or in fibrotic reaction to devices or microencapsulation technology. Maybe you can just halt or prevent fibrotic reaction to biomaterials or to systemic disease in which fibrosis is part of the pathology. Well, if it is a fibrosis secondary to cell death or liver injury, that then you have a fibrotic reaction, maybe the effect of the exosome is more to protect the cells and decrease the cell death.
Yeah, thank you for this suggestion. Yeah. Hello, I have a question. Great presentation from all three of you. It was very informative. The one question that I have, and this is commonly brought up when you're talking about cellular therapies, is the optimal cell concentration or dosing to use in those type of therapies. I know we're at the very beginning of looking at potential applications for exosomes, but what are your thoughts on the dosing or quantity of exosomes to be used in different clinical applications?
Okay. I will try to answer your question. This is very interesting, very hard part at the very beginning. So far I didn't mention there's going to be a question. So nobody asked me what the dose we use to do the therapeutic. And actually, I already calculated. I would say 5 mice serum exosomes can cure 1 mouse for the whole 2 weeks therapeutic process. So this is a really big amount of exosomes getting into the mice. So at the beginning when we use it, we don't care about the dose. We just give them the maximum we can give them. And then when it's come to really the human being, it's impossible. And so it's going to be another way, a source to get the exosomes. So we're trying to get the cell culture, the exosomes, and we also, there's in vitro studies trying to have a dose response curve and to see the dose, the accurate dose, and then the contents, especially to get the best function to the cells. That's still in vitro. So come back to in vivo, there's still the hard part by the end to really find out the dose back to the animals or back to the therapeutic. Thanks.
I think another question in the delivery of cell products is, you know, when you cryopreserve them and reconstitute them, how many are actually truly viable? And we hear all the time, you know, our cell product has 5,000 or 5 million, you know, cells per cc, and it really doesn't mean a whole lot. And it probably is you know, when you marry these cells to a matrix, it probably is an exosomal function, but what the optimal dose is really not very well characterized.
Let me raise a question as well. And I think it's very important when you look at the dose response. And we heard Dr. Ricordi speak to, you know, being able to doses, finding adequate engraftment. When we look at the model in the American treatment of disease, much of the time it's just symptomatic provision, you know, with palliative type of care. Does is there an impediment to developing an engraftment type technology, or will we continue to use an exosome base on a dose response base, and will we be guided by regulatory oversight to actually maximize the cost of the care as opposed to implementing a long-term solution to a problem? And I wonder, would the panelists would just speak to those points. Dr. Temple, in the point of translation, do you translate a cure or do you translate a value in the market for further development?
Well, one would hope you transfer efficacy and value, but not value to the market, value to the patient. So I think that, you know, you have to be introspective and really guided by a lot of clinical trials and basic science in deciding, you know, what really is of value to the patient, and really what you're trying to do. I mean, if we're trying to cure cancer, that's a pretty lofty goal, and something that, you know, may be beyond our scope. But if you're trying to basically deliver a product to mitigate the effects of cancer, that is valuable, but may fall short of the goal. So I'm not really sure exactly what we try to do when we market products, but certainly we have definable problems, and we try to mitigate those problems with products that bring some value. And I think that's the way I think about it. But thank you.
So in keeping in line with Doris Taylor's last slides of crazy ideas, I wanted to propose a crazy idea to you, because we transplant in the liver hundreds of thousands of microcluster of cells called pancreatic islets, which contain like 1,000 to 2,000 cells each, and you put 700,000 or more of this microcluster. That can continue to be a factory of insulin production now with results like with success at 7 years that is remarkable or comparable to organ transplant. And I was wondering, thinking about the microcluster of adipose tissue that could be a constant factory of exosomes, if there would be a role to test experimentally to put autologous microcluster of fat injected in the portal vein and see if they would produce exosomes that fight fibrosis in your model. Starting in a mouse, maybe.
Okay, we collaborate.
Great.
Are there any other questions that we may have from the audience in attendance? I mean, I'm full of questions here, and I'm going to go back to Dr. Temple again. You know, when we look at the diversity that you find of the markers that are specific to any tumor type, so if you're looking at let's say the chondrosarcoma is well differentiated, if you're seeing 5 or 6 different markers for that tumor, would it be reasonable to go back and implement the tumor style by using a common marker and then use the vaccine approach that you spoke to maybe attack it as a common piece? And actually you would really implement kind of almost a decoy where you would create almost a false tumor marker in those cells and then kill it.
Yeah, I think there are many, many strategies that can be employed, some that are constitutive, others that are more tumor specific. And again, I think that it's going to involve a lot of different strategies within a specific tumor type, within an individual patient, in order to effectively manage that disease. And we've seen that now, you know, with patients who've had conventional therapy, for example, adriamycin high-dose IFOS, and the tumor becomes resistant. And then they go on to the next checkpoint inhibitor, the tumor becomes resistant, and you keep developing strategies down the line, and ultimately you come to a dead end. So I'm not sure.
I think it's a good point, Judah. I mean, I think until we identify kind of the teleology, the strategy of the tumor cell to avert, you know, detection, you know, we're going to have challenges. And I mean, there are many ways that the exosomes are able actually to stimulate immune recognition as well, and I think those are great things. I don't want to carry this beyond the point of questions, and I would suggest we thank our speakers for just an amazing set of presentations.